Thanks, Katerina. I will also briefly share some of my personal history because it's the topic of the day. As Peter and Katerina, who both presented this morning, I was also born in Germany. I was fortunate enough to be born a little bit closer to the sea than these two. From Hamburg, I initially got into diving and marine conservation because I think, like many young people of my generation, adored Jacques Cousteau, and so I watched Jacques Cousteau movies, so I must admit I was initially a Mediterranean fan, and I only coincidentally came to Australia, to Lizard Island as a volunteer and fell in love with the Barrier Reef, and hence I'm still here working at Ames. So I'm going to talk about a CSIP project we're running at Ames, but also a lot of in collaboration with Morgan's team, so some of the topics I'm going to talk about may sound a bit repetitive to you. So we're using eDNA as a new monitoring tool with the same aim Morgan has to detect really early stages of outbreaks. So as I mentioned, it's a team of us at Ames in collaboration with Morgan's team and also with Emma and people at CSIRO. And it's CSIP project C03. As Morgan alluded to, it is assumed that the outbreaks start in this outbreak box, but we know now that it's possible that they start much further north. They slowly move down south, as they are now basically in the south of Townsville, and we have an ongoing permanent outbreak in the Swain regions. However, there's now indications that these are further north. As also all of you know, there's an active culling activity, huge culling activity at the GBR run by Cabrera and the operators. And the main idea of CSIP is to support these culling and optimize the culling activities and the management of the COTS on the GBR. I don't have to spend much time on explaining manta toes again. It is, as Morgan said, a good method to document existing outbreaks, but it's not fine enough to find early numbers. So in addition, low densities. In addition to the salad surveys, we had started at Ames to develop eDNA techniques to monitor COTS. eDNA is a technique, just in general, where you would catch free DNA in the water, which could still be associated to sails or could be freely in the water or absorbed to other surfaces, and you use various different techniques to identify those. In terrestrial and freshwater environments, this has been used quite frequently since the last 15 years for general biodiversity descriptions or detection of introduced species, but also in quite exciting studies on documenting the ecology of ancient species which are extinct, like the mammoth or the moa in New Zealand. There's a few different techniques which is usually confusing people a lot who are not in this field. One of them is meta-barcoding, which looks at multiple species and usually uses next-generation sequencing. This is a technique which gives you many species, but it's probably not, or only to a very limited amount, quantitative. However, what we do is single species detection with specific markers designed to only detect that species. And that can, at least to some extent, be quantitative. There's two methods, which is one is quantitative PCR, the other one we're using digital PCR. So you can quantify the number of copies in the water, and you can later on relate that to actual densities in the field, if you're lucky. We have, as I said, worked on this for over 10 years. We have already used this quite extensively. We have an ongoing larvae monitoring program, which is supported by lots of the culling people represented here. So most of the COTS control boats are collecting water and plankton samples for us so that we can monitor the presence and abundance of larvae. And we've used this for juveniles and adults. That's what we're going to talk about here. Peter Doyle, as part of his PhD, has used our eDNA technique to quantify newly settled juveniles on the reef. And as Pete Mumby related, we are also using this to look for predators in the gut contents of these predators. We're looking for identifying Crown-of-Thorns. But here, as part of CSIP 03, we're going to focus on adult and juvenile detection. Briefly, the workflow is you have to go out in the field. We're collecting 3 sites on every reef at the moment, taking 12 samples on these filters. So 2 liters of water are filtered over the filters. They're fixed in a fixative, and in the lab they're extracted and the DNA quantified. We had published 2 papers before we started the CSAP work. One was at really high COTS densities, where we showed a rough correlation between eDNA and COTS. The second one, just when this project started, we had looked at lower density populations, including some of the cell data at that stage, and we showed that the proportion of positive samples is quite well related to the actual density of Crown-of-Thorns, as depicted here. So the main purpose of this project, so the method basically existed. The main purpose was to test a large-scale monitoring, which we implemented on 16 reefs over the last 3 years. This monitoring was complemented by samples from up north. Morgan's team has collected as part of a cooperation with PO4, and we also later on added samples through ships of opportunity, specifically testing whether, for example, the Queensland Parks and Wildlife boats could collect for us and add to the data set. Jumping straight into the results, so the results can be shown like this. This is the first year of surveys we have conducted. You see roughly 20 reefs presented. The more red you have in those little pie charts, the higher proportion of positive samples you have. Now you might wonder, and I'm going to explain that later on, what does that mean? The more red, the worse, right? Any completely white pie chart has no COTS eDNA detected, whereas if it's completely red, all of them the whole pie chart's red. So you see in the first year, even up in Princess Charlotte Bay in Cape Granville area, we only had 1 reef which really had high densities, and which is the completely red reef there. And down at the south, you see the ongoing outbreak still documented at Davies and Bowden Reef, and high densities of COTS there. In the second year and third year, the number of red dots had quite increased, so the second and third year really high densities comparable to Morgan's data in Princess Charlotte Bay and in Cape Granville sector. Even the last year looked like it's getting a little bit less. You see how in the last year we really increased our sample size, which was not through increasing our own effort, but by adding samples other people like Queensland Parks and Wildlife have collected to us. And we also had a collaboration with Emma's group on calibrating these 2 methods, so that set another set of data there. So I'm hoping this shows with relatively small effort, you can investigate and test a huge number of reefs for Crown-of-Thorns in relatively fast time. Yes, the analysis does take us a bit in the field. Also, thanks to the collaboration with Morgan, we have now over 40 reefs where we can compare numbers we are taking with the numbers his team is counting in the field. We can derive a relatively good correlation between, or regression between, Crown-of-Thorns densities and our eDNA results, which means we can put this into practice and decide, for example, if we want to detect COTS at the 3 number per hectare, where would be the threshold be? Just for argument's sake, we say maybe we should say if more than 30% of the 40% of the samples are positive, there's an outbreak, which would mean, but these numbers are debatable. We could say maybe we just want to detect 10, so we could set that up, or maybe we want to have a more conservative estimate, so we set the number down. But just for argument's sake, that would mean we could translate the pie charty bars and charts into really easy numbers, basically just in a binary matrix where we can convey this to management, that we think on these reefs, which are depicted red here, are the reefs where some sort of management action should happen. Is it culling, or is it sending out more survey teams doing detailed salad surveys, or some mix in between? So in the end, a complex method, but it allows fast detection, and we can work on a lot of reefs at the same time and analyze, but we can distill it down to quite simple messages needed for management. Also briefly mentioning Lizard Island, because that was one of the, what we call a sentinel sites, similar to Morgan's team. We have data since 2019 from Lizard Island, and you see at the top graph that roughly at the same time when the salad data jumped up, the eDNA concentrations also jump up. The top line is the average of the whole island, whereas the bottom line is 5 different locations. With exception of the lagoon, which always has really low numbers, you basically see an increase in DNA in all 5 sites, which is really quite well corresponding to Morgan's data and the ones Josie Chandler has talked about before and already published. Uptake of the project, I think we are really closely collaborating with Cabrera and with CSIM and with the operators. We're annually reporting the data to Sam Matthew at Cabrera. They're all, the first 2 years are available in the COTS dashboard. They're used in a test case for an early detection system. So the uptake is quite well, and I think we conveyed well how these data can be used in the monitoring. Recommendations and conclusions. So similar to the salad surveys, we can clearly say the 5th outbreak has started. It is very likely that it started in Lizard Island, and it looks likely that it has started further north, as Morgan alluded to, which is really important to understand, because all the outbreak causes we are discussing, be it nutrients or predation, are really quite different if you're looking at an area further north. You could nearly argue both nutrients and predation should be less of a cause up there, but that's a case for further studies. Just making the point that we really need to know where these outbreaks are to start speculating or researching what's causing them. I hope I convinced you that the method is quite cost efficient and fast, and that with using ships of opportunity, we will make this even more efficient. It's by no way anywhere in competition with salad surveys. I think they're 2 very complementary methods. Our method could easily be used to measure several hundreds of reefs annually, basically, and then you could focus on the reefs where we recommend action with, for example, further salad surveys. The recommendations we will roughly spell out in our final report will be to continue this backbone monitoring on roughly 20 reefs, so that we can see how patterns shift over time. Also to have this monitoring complemented by ships of opportunity, such as the Joint Field Monitoring Program and other data. And we will likely also recommend to continue the Lizard Island data. As for Morgan's team, currently there's no funding to do that, so we're hoping to be able to do that a little bit over the next few years. And in addition to that, we propose to not stop the research and continue development. There's actually quite good uptake at the moment. I'm quite chuffed that even 2 publications from China are citing our method, and that these guys have developed what's called a biosensor based on COTS DNA. So basically just some probe you can stick in the water and get a yes or no answer. So that's obviously the next step, and we're hoping to take that on board and further develop our technique. Thanks.
Thanks very much, Sven. Are there any questions for Russell?
Okay, it's two separate questions, Russell. Firstly, yes, there's only there's two acanthester species in the GBR. One is brevispinus, and the other, the one with many names. It doesn't detect brevispinus, so it really only detects the coral-eating COTS. However, it would detect the other COTS from the Indo-Pacific area. To look at sea cucumbers or any other species, you would have to redevelop new primers and revalidate how they work, yes. But it is something we're proposing for a while, that it's used for a larger monitoring.
How much work is developing new primers?
Sentence with a piece of string comes to my mind, but I can't. It could be a year of a technician's time, yes. It's not, ideally it's been done for multiple species at the same time, but it is, with cross-test, it's more than a master's, put it that way. Or maybe it's a master's, roughly, year. Anybody else than Russell?
Lena?
Hi, Sven. Thank you. That was very interesting. I'm wondering what the spatial reach of this method is. So if you take a sample in very droplet research and you detect certain density, how can you know that's for a rough research or nearby reef?
It's a good question, and it's something, thanks Renata, something we're looking at quite intensively because we're getting it asked a lot. In the moment, we're statistically analyzing how well our reef-wide data match to the salad data, which I think I showed matches pretty well. If you look at locations, that's getting a little bit wobbly. It's not as great. So I think it's a reef-wide estimate at the moment. However, we have done transects across the reef starting from a kilometer windward to a kilometer downwind. Practically windward, we're not detecting any. Then we're hitting the reef like Davies, you get a signal and you get half a mile behind the reef. You might still get a footprint, but it's pretty quickly diluting.
Okay, I think we're out of time.
Thanks.
Thank you, Sven.